Thank you.
Hello, good morning. Hello.
Yeah, good morning, Dr. Soha.
Okay, you will share my screen or you will download it?
No, we will be doing that.
Okay.
Okay.
Can you see the screen, Dr. Soha?
Oh, yes, I could.
Yeah, thank you. Thanks.
Okay. Good morning, everyone. And thank you for inviting me for this conference. I'm talking today about the precision medicine and its future. 1 second. Okay. There is older concept of precision medicine, which was personalized medicine. The personalized medicine is almost an old term. It was hundreds of years ago. It was developed according to the development of chemistry, histochemistry, and microscopy. And for that, we have more understanding of the disease and its treatment. There are more than the term of gene sequencing have been a great leap in science and leap in precision medicine also. Okay, can I have my next slide? Okay. The term personalized medicine describes providing the patient with the right dose at the right time, at the right for the right person. No, I need the first slide, please. Okay. Okay. Personalized medicine is like tailoring the drug for the person according to its needs. It may be used for its protection and diagnosis and treatment. Okay. Next. Can I have the next slide? Okay. Okay, so there is an overlap between the precise medicine and the personalized medicine. Personalized medicine is have the accurate treatment for this disease. This is precision of the disease, while personalized medicine is the tailoring the treatment according to the person. Precise medicine is developed according to the pharmacogenetics or pharmacogenomics, which is science science between cooperation between two sciences. Is the pharmacology, the science of drugs, and genomics, the study of genes function. This study have been developed to have an accurate development of effective and safe medication and accurate dose. And this is varies according to from person to person according to its genetics. Okay, next. Okay, in this figure we can see the old concept of medication and the precise medicine. The old concept of medication is one size fits all, and this is not right, because as we see, we diagnose a person to have a certain disease and give him the certain treatment. This certain treatment may have three outcomes. One of them, it's okay, it will treat most of the patients, but some patients doesn't respond to this treatment or even have adverse effect. According to precise medicine, which is figure number 2, according to the patient's genomics and its DNA sequencing, we give the right medication to the right person at the right dose. From the precise medicine, we can avoid allergic side effects, and we can also have the best results without suffering from resistance of the drug. Okay, next. Okay, the prospect of applying precision medicine why we and what is the obstacle we have to have in precision medicine. This first of all, why we should have precision medicine and why it's developed. Nowadays, we have a large-scale biological database, such as the Human Genome Sequencing Project. And the sequencing have been developed, its cost, price, and its efficiency according to the time. Patient characterization also, which include the proteomics, metabolomics, genomics, and cellular assay and mobile health technology. And finally, the development of computational tools which allow us to analyze this large data and give a feedback to a good outcome for the patient is a good result of our development. Okay, next. As we know that cancer is a disease result from accumulation of mostly genetic damage, and part of the cancer is inherited genetic disease. So understanding and the risk and the diagnosis and the right category of the cancer and therapeutic strategies is a target for us for treatment cancer. Drug and antibiotics have been used for treatment of cancer, but still it's a huge obstacle for us to have a good treatment for cancer patients. So precision medicine is a good method of treatment of cancer patients. Can we have the next slide, please? Okay. So what we have in cancer patients, we can have unexpected drug resistance. We have genomic heterogeneity of the tumor. We can have also insufficient monitoring of response of the drug, or even tumor recurrence and tumor metastasis. Also limited knowledge of drug combination. This is the obstacle we have for treatment of cancer patients. Next slide, please. Okay, can I have next slide? Next. Okay, so according what we do in precision medicine for cancer patients, patient enters the clinic in day 1. Then we have a tumor biopsy from the 2nd day or the 4th day. We have also pathology review to know the type of the tumor. Is it malignant? Is it benign? Then we, at the 6th day, we have sample preparation. Then at the 7th day or the 8th day, we have molecular analysis of the tumor according to NGS new generation sequencing. We have now large data from the pathology and biomarkers and molecular analysis. We use bioinformatics to analyze the data and know the molecular outcome of the patient. Then the molecular tumor board assembly and decide which is the best treatment or protocol for the cancer patient treatment. This is a typical precision oncology workflow in the hospital. Okay, next. Why we use precision medicine for tumor patients? After molecular profiling for the patients, we have we have 3 outcomes. First of all, prognostic markers. Then we can determine drug sensitivity or resistance for the patients, and markers for prediction of adverse effects for the patients. According to these 3 markers, we can determine which is the best treatment for the cancer patient. Okay, so what is the clinical implementation for the patient? First, the patient we take blood sample and tumor sample. The blood sample, we can will lead to having serum glycans, microRNA, DNA, and interleukins and other biomarkers, and we can determine genotypes of the patients of the tumor. The tumor itself may be solid or liquid. According to the tumor, we have sample of it, and also we can have MRI for the tumor, or MRS for the tumor. Also, this is in vitro non-invasive methods. Some hospitals make xenografting for the tumor itself, take a sample from tumor and put it in animal like mouse, and try to treat the mouse by a different method to see which best medication that will lead to healing or removing the tumor from the animal. After having the blood sample and the tumor sample, this will lead to have a classification of the tumor. Is it malignant? Is it normal? Or it is pre-invasive? According to single level classification, we can determine it by X-ray or MRI, but we have another classification according to histopathology. It's a classification of pathology-based classification, and also genomic pathology according to the methylation of the sequence itself. We can have also the transcriptomics classification, which according to mRNA or non-coding mRNA, non-coding RNA or microRNA. Also, we can analyze the proteomics of the patient, the proteomics including protein, meaning the protein's expression of the cancer, using LC or mass chromatography or MS way. We can also can analyze the metabolomics of the cancer itself, metabolomic expression, using HR mass or MR classification. According to this, all of this, we can have our precise medicine by analyzing the genomics and transcriptomics and the proteomics and the metabolomics. And I may add also, there is microbiome analyzing for the patients, because there is interaction between the patient lifestyle and its genomics and the cancer itself. So precision medicine is not just the disease itself, but also his lifestyle and the microbiome and his habits in eating and so on. So precision medicine, after all of this and big data analyzing by bioinformatics, we can have response signature for the treatment, or resistance signature for the treatment also, and adverse effect signature for the treatment. This is for personalizing an accurate treatment for the patient. Okay, next slide. Okay. So current emerging molecular approach enabled for precision medicine this is integration between precision medicine and other diagnostic clinical practices. One of the diagnostic clinical practices are using immunohistochemistry. This is one of them, using immuno-oncology marker like BDL1 expression for immune checkpoints, and also using FISH, which is fluorescence in situ hybridization. This is also one of the golden methods for DNA rearrangements with the NGS to have a full characterization for the tumor. Also, there is a debate between using NGS or whole exome and genome sequencing, because NGS using only a single gene testing for a certain panel for disease. Not all the genes are tested, while the whole genome is using the whole DNA for knowing the patient's itself DNA sequence. Also using circulating markers, biomarkers, including circulating tumor cells and circulating DNA of the tumor and circulating RNA and protein for the tumor. Okay, next. So how to expand the precision medicine? With the time, we'll have a bigger database, and we can connect each disease with some mutational signature. This is by knowing which that disease have certain mutation at certain points of DNA, or also knowing the gene expression signature for the disease. Okay, next. Okay. So we have near-term goals and long-term goals. The near-term goals, of course, the innovate clinical trials use a combination of therapies to give the best results and know how to overcome our drug resistance, and knowing also to how to overcome the tumor metastasis. Okay, next. Okay, longer term, according to after a longer time, I think we'll have to advance our pharmacogenomics, and it will be cheaper for the patients. Also, identify new target for treatment and prevention. Also, using computational devices and mobile devices for enhance our patients' healthy behavior. So, and also making a scientific foundation for precision medicine in many diseases. Okay, next. Okay, thank you so much. Thank you.
Thank you for your talk. Any questions for Dr. Soha?